Hi, everybody. My name's Ann Sharuti. I am an associate professor of pediatrics at Emory University School of Medicine, and I'm also an affiliate scientist in micro and immuno at the Yerkes National Primate Research Center. The work that I do primarily focuses on using the non-human primate model to test cure-directed strategies and investigate HIV persistence. And we focus, on both adult monkey models, but also are quite interested in sort of applying a lot of what we're learning on the adult side to a pediatric or infant monkey model. And nice to see everybody on the screen.
Thank you. Next we have Brad.
Welcome.
Hi, everyone. I'm Brad Jones, Associate Professor at Weill Cornell Medicine in New York. My research focuses on trying to figure out how we can harness CD8 T cells and NK cells to deplete the HIV reservoir. And that work has really led us in a direction recently of thinking that latency may not be the only barrier that stops these cells from killing the reservoir. And we're really interested in this idea that the cells that persist might have been selected for intrinsic resistance to being killed by cytotoxic T cells, and developing strategies to overcome those mechanisms is something that we're particularly excited in pursuing. So thank you for the invitation.
Thank you for joining us. Luis.
Yes, good afternoon, everyone. From Philadelphia, I'm Luis Montano. I'm a professor at the Wistar Institute, and together with the partners in our community and different academic centers, we are overseeing the Beat HIV program. Our emphasis has been on harnessing innate responses as well as adaptive responses through gene therapy. So we have clinical trials underway, and our emphasis has been in to try to monitor how to interface community and research as we go along this journey. So thank you for the invitation, and I look forward to the discussion.
Thank you. And is Josephine, has she managed to join us?
Yeah, unfortunately, it looks like not being able to make it. At least, yeah, I'll keep an eye out.
Okay, great. Well, we'll just start with our three guests, and thank you again to each of them and to each of you for joining us on today's call. And so I would love to start off with having Danielle have the privilege of giving us the first question. Pitch the ball.
Thank you. Give me 1 second as I switch screens. Hold on, please. I'm muted. Sorry. Hi. Okay, I'm back. So our first question of the day, and so the questions will pretty much run in sequence. Any of the panelists at any time can feel compelled to come off mute and answer, and we appreciate your answers. In response, Michael, did you give the direction to folks to either type their chats in the comment or? Okay.
I did, and so we will make sure that they're reminded of this, that again, if you want to ask a question, just raise your hand and we will come to you so you can ask it live. Or if you want to type it in the chat feature, we'll be monitoring that way as well. So we have two ways of you asking a question, both of them acceptable.
Okay. Oh, Linda D has a question.
There we go.
Hi, yeah, I thought they were going to give us an update, and then we were, on what they thought was the most important, and then we were going to ask them questions. I think that might be why you're not hearing from people. But I have to say that I was intrigued by what Brad just told us about resistance. That, oh God, I hope it's not true. But anyway, another obstacle, right? Maybe he can tell us what he, the most recent knowledge he has about that. Other obstacle to HIV cure.
There you go.
Potential obstacle.
Okay, so yeah, I think there's a few lines of evidence that are converging to support this idea that cells that get recognized by CD8 T cells or NK cells don't necessarily die. And I think the field has been thinking about this for a long time from the perspective of the CD8 T cell or the NK cell. If this doesn't kill the target cell, it must mean that the CD8 cell isn't strong enough or the NK cell isn't strong enough, and we need to come in with therapeutic approaches to make those better killers. But I think the other side of the coin here is that target cells are not all the same in terms of whether or not they will die if they receive the same inducement to die, the same cytotoxic hit, we say, from a cytotoxic, from a CTL or from an NK cell. And so that raises the possibility that the cells that live for a long time and go on to form the reservoir might have been selected for these cells that are better able to survive T-cell killing. And I think one talk that stood out to me from this conference that I thought was particularly important in this regard was from Kevin Einkopf, and I think he's from Matthias Lichterfeld's lab. It might be maybe Zhu Yu's lab. I'm not entirely sure, but they rolled out a new technology which has the pretty amazing ability to capture both HIV proviral sequence along with the HIV integration site, along with whether or not that exact provirus is making HIV RNA. So is it completely latent or is it expressing itself? And they find that while over time on therapy, there's a general reduction in expression from RNA, as you would expect, the expanded clones, some of these continue to make a lot of RNA and even to increase the amount of RNA that they make over time. So this raises the question of why these aren't getting recognized and killed by the immune system. And so then I would also mention, since I was asked the question, RTOC, which has gone in to look at some of the mechanisms that might enable these cells to survive killing. And what we've really been doing, this is all in vitro assays. So we set up kind of optimal conditions where we think that the killer T cells should be able to kill the infected cells. They kill many of them, but some survive. And then we can ask what's different about these cells that survive. And we found a number of mechanisms so far. And I think an exciting aspect of that is that some of these might be targetable by drugs. So the first is a kind of a master regulator of survival called BCL2. And there are BCL2 antagonists that are clinically approved. And the most recent one, which we presented, is a gene called EZH2, which can limit the ability to which these infected cells are presenting the virus to cytotoxic T cells. But I think, Linda, to kind of your point, we could think of this as yet another barrier, and that's kind of like the pessimistic, I think, approach. Like even if we overcome latency, now we also have to overcome this resistance. Sorry, there's a bit of background. But I think the other way to look at it is, since there's this evidence suggesting that...
Brad, could everyone be sure to mute? I'm going to mute everyone. Just because we have some background noise. It sounds like a TV is on here. And then Brad, I'm going to unmute you. Just so, or you can unmute yourself again, Brad. Let me see. There we go. Oop, ask to unmute there.
Oh, great. Unmuted again.
Here you go. Let's do
that.
So, there's something going. It sounded like...
Yeah, so there's two ways to look at it. I mean, we could look at it, okay, now we have to overcome latency and these barriers to killing. But I wouldn't suggest looking at it necessarily that way because again, there's this evidence that some of the reservoir is continuing to express antigens in individuals on long-term therapy. So that suggests that even if we overcome the resistance by targeting some of these mechanisms that I mentioned or others, maybe we can have an impact on what we're now appreciating as an active reservoir, kind of alongside the latent reservoir. So I don't think that that all of the obstacles necessarily have to be overcome at the same time to have some impact, even though to get to the eventual goal of eradication, we probably will need combination approaches.
And so, Brad, we had someone ask a question about the group that you were speaking of. I think it was before what you just mentioned. Corrine wanted to know what was the name of the group.
Yeah, so the presenter's name was Kevin Einkopf. I think I'm not sure if I'm pronouncing that right.
Matthias's team.
Yeah, he's with yes.
Can I ask you, Brad, about the drugs that you mentioned that are already approved that you might be looking at? Can you just tell us a little bit about them? I mean, hopefully they're not, repurposed cancer drugs, or they Do they come with a lot of toxicity like we've experienced with the other sorts of drugs we've tried, or is this going to be easier, you think?
Yeah, so obviously an important question and consideration. So I would say that for the BCL2 antagonists, so ABT-199, venetoclax, the side effect profile is considerable. I'm not a physician, so I wouldn't want to say it with too much confidence, but I think that there definitely be careful considerations as to whether or not that's something that makes sense to test in the context of otherwise healthy people on antiretroviral therapy. So that's why we're continuing to push down that road to identify new hits. So our new hit, EZH2, there's EZH2 inhibitors, which are approved drugs that have less significant side effect profiles, at least, than the BCL2 antagonists.
Mm.
We haven't really explored with physicians yet whether or not that seems to be a viable thing to test. But I think we're just barely scratching the surface here. In the screens that we're doing, we're getting hundreds of hits to sift through, and we're trying to prioritize things that have a potential path to the clinic. But I think as we kind of go through that, I hope that we'll come up with some hits that are selective enough that we can try them without an extensive side effect profile, and or use different types of targeted delivery systems to get them to just the cells that we're trying to sensitize to killing.
So let's move on. I wanted to hear what Anne, what did you think was the most consequential or the most important bit of cure-related research that you found at Croi?
Well, that's a tough question to answer, but I think one thing was clear, just to say it at the outset, is that this meeting was, not all HIV focused. There was an awful lot of COVID research that was presented, for better or for worse. And so, clearly we all know that a lot of HIV clinical trials were put on hold as a result of the COVID pandemic. So, that I think led to sort of a more limited array of studies being presented. That said, just sort of focusing on the pediatric side of things, because that was sort of my charge for this, although there were some other really, I think, interesting talks that I'm happy to bring up as well. There were some studies that are sort of diving into characterizing the reservoir in perinatally infected infants and children, and even adolescents, folks that are and older that have been infected since birth, and looking at levels of provirus, kind of characterizing them with more sophisticated assays than really has been done before. So I thought that was nice to see from a couple of different, one from the KARMA cohort, that's a bunch of different European centers, and then another from a cohort in Mozambique. And so, basically what both of those studies have shown, and I think a lot of other people are looking into this as well, that the levels of intact provirus in perinatally infected children who have been now on long-term ART are extraordinarily low, which on the one hand is, exciting, which means maybe they're more kind of amenable to a cure approach, that there's sort of less reservoir to target. On the other hand, some of it may be just a consequence of, the assays and blood volumes and the things that we all struggle with in pediatrics. But the flip side, I think, is that when we're thinking about doing pediatric clinical trials, if you're already starting from such a low level, it's awfully challenging to think about how you would measure an effect of your cure intervention, because it's hard to go from undetectable to lower than undetectable. So that's just something that I think was hit home by a couple of the different presentations. I think in terms of the studies that were non-pediatric focused, that were actually trials that were conducted, there were a couple that were presented. One was from Joe Casaza from the VRC, and that was, I think, the first time I've seen it presented at a conference, but maybe it's been presented earlier. But this is a very small study, but it is of adeno-associated virus-mediated delivery of VRC07, the broadly neutralizing antibody. And it was only 8 volunteers, so really small. But they did show that, I think, sort of for the first time in humans, that this approach could actually lead to expression of VRC07 in a person. And it was pretty well tolerated. They said they had no adverse events. And the kind of main limitation was that subset, it's already only 8 individuals, so even smaller subset of those people did develop antibodies against the VRC07, and so that limited the expression in those people, which is definitely a known issue with this approach. But it's something that I think, there've been a lot of monkey studies, or there are a lot of monkey studies that are going on with this kind of AAV delivery of various broadly neutralizing antibodies or other like ECD4, other kind of approaches that are designed to reduce the reservoir size. But this is the first human study, I believe.
Thank you for that. I wanted to move on to hear Luis's answer for the same question, though. Like, Luis, out of all the things that you saw at Croi, related to cure in some way, which really stands out to you?
Thank you. So I think, last year was about intact versus defective, right? Last year, we were all wrestling with the new data and the new assay from the Silicano lab, allowing us to detect intact. So we were all very hopeful that this would be the path to measuring the right amount of the amount of residual reservoir that we needed to go after. And I think this year, the biggest news is location, 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 meaning that it's not about the intact alone. It's about where it sits. And we need to now target the ability to measure which of the intact proviruses we need to be worried about. And there is some evidence in the elite controllers that the location may be misleading, meaning that you may have a lot copies and still control, which obviously we always view that amount means no control. Right. So they are telling us that the location may impact how you may interpret the amount. Then the other side, which Brad noted, was that, well, if we always expect the immune system to be controlling what it can see, how come we can have expression without seeing it? Because this individual has control, but it is expressing RNA. So is this what we before used to think about, a small smoldering antigen amount or expression that keeps the immune system able to control? Or is this a red herring that basically has no impact whatsoever? And so this type of information, I think, has raised some different perception of what it means to measure intact. And second, what would the right immune response needs to be when the virus is being expressed? So I think for me, that was the biggest news, because it basically impacts every cure study that you can think of into how we need to be evaluating the impact of the strategy. Right. Because if we then go, okay, well, we're just going to measure the amount of intact provirus, then how do we know that we're not being misled by measuring the intact provirus that is quote unquote dead, meaning that it's not being expressed? Right. So I think that this dynamic is going to change how we are going to be probing the assumption of measurement versus clinical control. So we still know that clinical control rules, right? So that is what rules in the end. So, but the expectation that the intact was going to give us a straight shot towards predicting control, I think that to this year, that idea was out the door.
Right. And in my opinion. Location, you were not talking about location like where it is in your body. You're talking about specifically where it's located
in the nucleus.
Yes.
Because, you know, the virus comes in and it kind of fits into your cell's nucleus. And
Right.
we thought that once it got there, it meant that it was fair game, that it could be expressed.
Right.
And now we're learning that depending where it comes in, you can either express or be a dead end, but still have an intact provirus. So then the question is, how do we start teasing that apart in measurements that would not otherwise be looking to determine where it's located, because we just were measuring the total amount.
So this is related to the famous Lorene Willenberg case that came out, where they were trying to capture what was happening to her, but it's really where HIV embedded in her DNA that made her become an elite controller.
Correct.
And basically,
Correct.
have such
Correct.
a low level of HIV because of that. It's all where in the DNA HIV embeds.
Correct. And I think that dynamic as to what makes it go one place versus another is a new opportunity to determine how do we go after it. Because now we have to go after the one that is the problem, not necessarily the ones that are dead ends. So I think that's going to be some, new area that we should see some activity on. I think on a more strategy side, the presentation that I thought was discouraging was the fact that the results from Badam Baruch, last year that were really exciting about non-human primates that were given vaccine and BNABs, and they got control, and we all thought, well, that means that the BNABs could be a segue towards remission and control. They reproduced that in NIH, and they didn't see the same outcome. They didn't see any control. So that then raises the question of, is it the model, is it the type of antibody, but it's not going to be a sure shot, meaning that whatever BNABs you add and a vaccine, it means that you're going to get control. So that was, I guess, something that I was disappointed to see, but it's the way it is, right? You get one result in one place, then you repeat it, and then you learn something new.
Right, which is something that we always have to counsel people, because, everyone's very quick to publish results sometimes, and lots of institutions often trumpet the results, but when they haven't.
Well, but what I'm saying is that it could be, I'm not suggesting that the research was wrong.
No.
Because the question now is what was different between the approaches and what led to one versus another.
Exactly.
But then that qualifies the first result, right? That doesn't say, like, for example, antiretroviral therapy. We know that we give antiretroviral therapy to everybody and the virus comes down. And there's not the expectation that it will go down on you, but not on this other person.
Right.
So now we have to wrestle with, well, what was different and understanding between both. So
it looks like now, Linda D has a question for you. Linda, did you want to take yourself off mute?
Yeah. Well, that DNA issue is, I guess, in other words, do we know what challenges you will face on one part of the DNA as a result of the other? Can you explain a little bit more about the different locations on the DNA? I really don't know anything about that. And the other thing is, how hard is it to pinpoint what spot, is involved? And I guess, what is it harder to do? Will it be harder to deal with in different spots? I mean, you may not know the answers to this stuff yet, but that all occurred to me as you were speaking. Thanks.
I mean, I think all the questions that you're raising are the ones that are being asked moving forward, right? How do we deal with the different locations, and are they targetable or not? So, but to kind of give you one example, I don't know if you've seen the cartoons of chromosomes that are like an X, and they have like a little dot in the middle. I don't know if you've seen those types of illustrations.
No, sorry.
But anyway, let's assume that you see an X, and then the dot in the middle is usually called the centromere because it's in the middle, right? For centromere. So an integration that is within that area is transcriptionally silent. So that means that if it goes in there, it basically is not able to be read as RNA. So it's basically a dead end. And that is one of the regions that was described. There are others that are, when we talk about transcriptionally silent, it means that the virus cannot be read. And as a consequence, the DNA is there, but you can't read it. So that's what we would refer to as a dead zone, or an area that is not able to make more viral copies, even though the DNA sits in the genome itself. Does that answer your question? Without getting exceedingly technical? Or Brad,
Yeah,
you
well,
can,
no,
you can
it's
give another
helpful.
shot at this.
It's helpful, and we'll learn more as we go. It just what occurred to me, and I thought you may have, but you can keep us posted.
But basically, they described like 3 different types of regions, all of which were quote unquote silent. And the one I was telling you in the middle of the chromosome is one of the 3, but the others are kind of more technical. But the bottom line is it's not being read. It's like a block and lock type of situation.
And I think if I'm thinking of the same talk, the kind of one of the concepts was that if HIV is integrated within a gene, it's more likely to be able to express RNA. But if it's not integrated, if it's in an intergenic region, it is less likely to have to be expressing RNA. And so that may be, I mean, that's probably oversimplified, but that's a way to think about, which of those 2 different types of intact DNA may be able to be actually induced and replication competent, and sort of, which ones, the ones that should be induced and replication competent, obviously those are the targets for a cure strategy. But on the flip side, as Luis just said, and I think is coming up in the chat, is that maybe if we're able to kind of induce the suppression in the block and lock approach, that could be, maybe there's 2 different approaches for the 2 different types of reservoirs.
This is Mark Milano. One quick question. This is all new to me too. Was the news at the conference where it integrated, or the fact that it can integrate at different points? Because I never even knew it could integrate at different points. Has that been known for a while and they just delineated where it does it, or is it news that it can integrate into the DNA at different points? Because I never heard that before.
Yeah, the sites of integration have been known to be very diverse for a long time. Essentially, HIV prefers to integrate into genes, and genes that are actively transcribing usually. I think what's new is the evidence showing just how much the immune system or other factors can select and get rid of some of those proviruses that can be expressed, leaving only kind of this, in some cases, in extreme cases, this these almost fossils of viruses. The provirus is still there, but if it's inserted somewhere, as Lewis was describing, that where essentially the lights are off, it's an inactive part of the genome, nothing's happening there, then it may never make a virus. And the other way I think about this sometimes, I don't know if this is helpful or not, but we know, for example, that there's CD4 T cells and there's CD8 T cells. The CD4 T cells still have the gene for CD8 in them, but they never express that gene. It doesn't matter. You stimulate them you treat them with whatever you want in vitro. Those CD4 cells are not going to start to express CD8 because that gene has been permanently turned off. And so if HIV is in a place where it's similarly turned off, then we probably don't have to worry about it. But as Richard nicely explained in the chat also, there's a big gray area in between. There's some sites of the genome that are very active. There's some that might be completely shut down, and then there's a whole intermediate range that we need to grapple with how to deal with.
I guess another way to look at it is before, you know that Finding Waldo books, where you have to find Waldo in a huge picture.
Yeah.
So before, we were not able to find Waldo because we didn't know which was the intact provirus. And then we were just blinded by the whole group. Now we know that how to find them, how to find Waldo. But now the problem is that some of these are in locked rooms and some of them are not in locked rooms. And what they described in this finding was that how to identify the locked rooms and the unlocked rooms. And what we knew before is that the integration could happen anywhere. What we didn't know before is that once knowing which ones are the Waldos, that they are in locked rooms as well. And that's the part that's new.
Thanks. That's really helpful.
So can I ask you one thing, Luis? The locked rooms is it good to be, is that like a block and lock room? We don't have to worry about them. We just have to worry about
Correct.
the one in the
Correct.
unlocked rooms, right?
Correct.
Perfect.
Correct. But
Great.
but the irony, or at least the complexity, is that some of these locked rooms have the door open, but Waldo can't get out. So the RNA still gets expressed, but the virus doesn't go anywhere. So that's what Brad was saying, that there's some of these sites that are still expressing RNA, and yet there is control. So then the question is, why is this not getting rid by the immune system now? So there must be some other layer of evasion, if you like, or lack of ability to see it, that is allowing this RNA to be there, but yet not be a problem for the individual.
So might this unlock the block and lock? It's sort of approach. I mean, we're kind of stuck with that now. We know it sounds good, but we don't know how to do it. So might this elucidate?
But I mean, one word of caution. What we don't know is that these are very unique people that were described, right? So we don't know whether we're chasing a calico cat or it's actually something very common in most people that are living with HIV and under therapy. So now the focus is, now that we know these types of questions can be pursued, let's go into the majority of people living with HIV that are under antiretroviral therapy and see how that type of profile reflects itself in these types of settings. Because what we were talking about is those very few individuals that are able to control the virus without therapy. And they're called elite because they're very, very unique. So the question is, how do we then try to move that into the majority of people?
And so do you know, like, what are the next steps that are being worked on with what has been so far learned research wise? Do you know what's being, is anyone pursuing these things that you've mentioned and the questions that we still have to answer?
Go ahead, Brett.
Maybe I can jump in there. So I think we have a lot of exciting new assays, and it's really about bringing these together, and a lot of people are doing that. So to just mention one example, there's this assay HIV flow that Nicholas Chamont's lab has been presenting, which lets you reactivate cells ex vivo and see those very few cells that are able to make HIV protein. And so you can sort those, for example, and then use some of the other cutting edge techniques developed by other labs, such as MIP-seq. And what MIP-seq does is let you look at the whole HIV DNA sequence, as well as exactly where it's integrated in the DNA. So now you can bring those three pieces together. This is what the HIV looks like itself. This is where it is. And we know that it was able to get out of the door, as Lewis put it, because that's how we identified that cell in the first place. And so there is, and to mention one other, to highlight one of the scientific spotlights, there was a talk from Guinevere Lee, who developed a new way to look at the RNA that we're picking up and then ask, did that RNA come from an intact provirus or a defective provirus? So now we're able to see Waldo at a different level, at the RNA level, in addition to the DNA level. So I think there's a lot of work to do to bring these things together towards a really comprehensive understanding of how the location in the genome influences the degree to which we need to worry about these proviruses. But I think the exciting thing is we have all of these new tools, and we're always hearing about new tools. It's hard to keep track of all the different SEEK acronyms that we have out there. So the technology is there. I think it's going to be an exciting journey to sort all of this out.